Hello, students. Good morning, good afternoon. I will ask you to please change your names to your team name and then your name, just so we can have a better control on who is here. And we're going to start our judging session soon.
Hola, muy buenos días.
Okay, so as it's written, this is room 1, group 2, health. The first team is Fénix. So if you are not in the right room, please leave and then come back in your time. We are going to keep just the people who are written in their names, Fénix, and then their names. The other people are going to be removed from the session. So Ale, I'm going to ask you to please roll their promotional video so we can start.
Sure, let's go. Can you tell me if you can hear the video?
We can't hear it.
No.
I believe you need to stop sharing. Yes, and sharing it.
Okay, I will try again. Sorry.
No, it's not coming through.
No, we still can't hear it.
Sorry, how can I enable audio? Can you help me?
Yes, I'm going to try it here. Hang on.
When you share the screen, there is an option down left which says share audio also.
Share sound. Yes, exactly.
Yeah, I can see that option. So can you try, please?
Let me do it.
I'm sorry.
That's okay. Share sound. Yes, exactly.
Yeah,
Nowadays, antibiotics come in all different shapes and forms, with one purpose: to stop bacterial infections from spreading. Over time, bacteria have become resistant to some of the antibiotics that used to have an effect on them. This makes them difficult to attack. Antibiotic-resistant bacteria represent a danger to humanity and are the cause of millions of deaths a year. It is believed that by the year 2050, deaths caused by multi-resistant bacteria will be leading over certain well-known diseases such as AIDS and malaria. One common bacteria that has some antibiotic resistance is the Staphylococcus aureus, which has become resistant to penicillin and other common antibiotics like methicillin. The resulting bacteria is called MRSA. It stands for methicillin-resistant Staphylococcus aureus. In Colombia, half of the intensive care units contain patients with MRSA infection. Antibiotics are compounds that inhibit the spread of bacteria in some way. However, bacteria can undergo random mutation within its DNA. Sometimes the mutation will result in a bacteria that is resistant to the antibiotic that was prescribed. These mutations happen when antibiotics are misused, such as taking too many of them or taking not enough of them. This is why we only take what the doctor prescribes us, none more, none less. As of right now, bacteria are winning this war. But all is not lost. There is a group of researchers that want to provide a solution to this problem.
We will use an ally of nature, good bacteria, which do not cause harm to humans or animals. We will give the necessary characteristics to this subtilis so that it can recognize the type of resistance of the bacteria. By detecting what resistance it has, the appropriate treatment can be administered in the shortest possible time, avoiding deaths from resistant bacteria.
So this is their project. I will ask the judges to please raise their hands so I can see who's going to ask questions. Yes, Margareta.
Thank you. So I want to congratulate the team to have chosen a really excellent topic for your project. It is a huge problem worldwide with antibiotic resistance. I have several questions, but I'm really curious about the human practice. And also if you can tell me a little bit about the antibiotic resistance problem in Colombia. Can you buy antibiotics without a prescription in Colombia? And I wonder whether the problem in Colombia is bigger than in other countries. Thank you.
Team, you can answer.
Okay, so Anderson, you go or I go?
Please go ahead.
So yeah, here in Colombia, you can buy antibiotics without a medical prescription. So that's like a really big problem because you can just go to the pharmacy and buy anything you want. And that's a really problem that nowadays it's being regulated, but you can still get them from any pharmacy. And in Colombia, there's also a problem, and it's about the geography of Colombia. There are places with it's really hard to get to those places, and it's really hard to assess the problematic. So you can't be sure how big the problem really is comparing it to other countries. But there are some other countries like Chile that have like this similar problem. But even if the problem is similar to Colombia's problem in Chile, these antibiotics are regulated. It's not like here in Colombia. So yeah, it's like part of the problem.
Thank you.
Also, the status of the resistance in Colombia is a little bit hard because we don't have a surveillance about the resistance. Actually, we have searched for some studies in order to know about the resistance of Staphylococcus resistance, and we didn't found a lot of information. We found like a study from 2003, so it was extremely old.
Do you have a follow-up question, Margaret?
If I can, yes. Thank you for answering that. I'm also wondering if Colombia has a public health organization which could help out with the surveillance and the information about the spread of antibiotic resistance.
Yes, Anderson.
Okay. Yes, here in Colombia, there are some organisms that we can found that use their power in order to the public health. We have here an organization that is called CVGILA. CVGILA tries to do a surveillance about the epidemic things that happen in Colombia. Also, we have the minister. I don't know how to say it in English. The minister. Maybe Sebas can help me.
Maybe the health minister. Like.
Okay.
I'm happy. Thank you.
Thank you.
I believe Pablo had a question.
Yes, if I can go next. I wanted to ask, how do you visualize the final product to happen? How do you, let's say that you did all of this in the lab, it worked. What would it be at the end? What would you do if you want to test if someone is infected with this?
Bueno, sería principalmente como un kit. Sería principalmente como un kit de diagnóstico que tendría como todos los compuestos para que ellos como que lo puedan realizar de una manera más sencilla sin tanto protocolo para que sea como complicado de observar el diagnóstico. Este sería como un kit, se transportaría la bacteria.
Can you please answer in English?
I will help my mate. He's saying that we want or we see our product like a little kit, and that kit is going to be a portable kit, so we can we can deliver this kind of diagnosis to the places that do not have the conditions to bear with the normal diagnoses that are extremely how do you say costoso? Expensive, yeah, extremely expensive. So that's the point, and we see like a little kit with all the parts that they need to work. It needs to work. Excuse me.
Like we were thinking this product to be like a point of care system. Yeah, because you see that like all of these complex diagnostic processes like lateral flow assays and PCR systems like being the gold standard, and we want like something more easy to use. Like as I was saying you guys before, in Colombia there is like this really big problem with geography. So there are like really isolated places where you can't take these kinds of expensive equipment, and also the personnel. We don't have in these places trained personnel. So this could be this could facilitate the process, the workflow process for these people.
Okay, I guess that we are running out of time, so maybe like a quick follow-up question. I think it's great about the kit, but what would you include in the kit? What will the kit be? Will be like the Bacillus subtilis at the end? Will be the bacteria? Will be what format is it?
Yes, we were thinking like aliquots with the transformed bacteria that has the circuit, and we were also thinking about we were like trying to decide whether we were going to give this in our circuit. There are some things we have to like put in the system, some how do you call it? What's it in sondas?
Rules.
Like probes. We have to addition some probes. We were thinking about additioning these probes, or we could make the bacteria to synthesize these probes in order to the test work.
Thank you. Emiliano, do you have a question?
Yes, hi, thank you. Everything was very good to read. I'm more interested in the part of the sequences because I don't know why it's the system like this, and will it not be more easy to purify the proteins outside the bacteria and then do like the test in the tube? Because I understood that you mix the something containing the DNA with the bacteria that is already in the agar plate. Is that correct?
That is correct.
Juan, I think you're muted.
We have chosen Bacillus subtilis because it has the capacity to sporulate. So we wanted to deliver this kind of diagnosis kit to places that do not or cannot have the normal diagnosis because they are extremely far away from the cities or that kind of things. So we want to deliver the Bacillus subtilis in a little tube, sporulated, so it can get it to that places. So that's why we are going to use the Bacillus subtilis with that circuit and not the proteins alone, because they are extremely unstable.
I'm good. And I have another one. How easily could you change, like in a plug and play system, to detect other organisms or other sequences in case the resistant bacteria, for example, have a mutation in the target site of the Cas12? I think you are using, not the Cas12.
It would be just Anderson. Do you answer or do I?
No.
It would be a matter of just changing the guide RNAs or the CDAs of the very same cassette.
Complementing the answer of my mate, we have seen this kind of things because we have some meetings with some doctors and that kind of person, and they said that it would be awesome to just not diagnose bacteria, but also, for example, fungus or other parasites. So we have thought about, for example, Toxoplasma gondii or that kind of organisms that are endemic here in Colombia. So we hope to get to that point, this kind of diagnosis.
Thank you. And the sequence that you are targeting with CRISPR, with the CRISPR system, is also responsible for the pathogenicity of the strain, or is something that some region that could have a hypermutation or be more variable? How did you choose that site?
Okay, so MEC1 is responsible for the repression of the resistance system. So it's always expressing. That's why we chose this particular gene. The other two genes, Anderson, was FEMA, FEMA4. It was a cytoplasmic protein, right? That was in charge of the pathogenicity of the bacteria. Would you go try to complement this idea?
Yes,
Thank
of course.
you.
FEM-A and FEM-B are proteins that are in the cytoplasm of the microorganisms and also help with the pathogenicity and the resistance and the construction or the building of the cell wall. We have chosen 3 because the normal diagnosis must be with 3 genes because it is difficult because these 3 genes are I don't know how to say, because there are like 3 groups and each group has 1 gene. So we must use the 3 genes in order to know if we have Staphylococcus resistant to methicillin, because 1 gene it can be another organism with 1 gene.
Thank you.
Thank you.
We have time to just one more
Congratulations.
question. Alejandro, please.
Thank you. So it's kind of a follow-up question, very short. So can you explain a little bit better how the detection will take place? Because you said that so the sample from the patient will be incubated with your bacillus. How will the genetic circuit detect the presence of this RNA? How will that happen? Can you elaborate more on that, please? Thank you.
So now our moderator will be presenting you like maybe an image, and what we want or what we think of how this would work is that so the cas in this cassette has a signal peptide that it's associated with the excretion pathway of the bacteria. So this excretion pathway is often used in industrial applications. So what we have in mind is that once the protein is expressed and once the protein is translated and structure is formed, this functional structure is formed, then the Cas, along with the guide RNA, are going to come out of the bacteria and through this excretion pathway, and they will, the recognition system is going to take place outside the Bacillus subtilis. Yeah, so that's how we have in mind.
Okay, thank you.
Due to our time, I'm going to stop us right here. Thank you, team. Thank you, judges, for the collaboration. If everyone in Team Fénix could please leave the room, that would be great. Thank you.
Thanks a lot.
Thank you too.
Thank you.
Thank you.
Bye. Bye.
Ale, can you please share the screen holder, please? So I'm just waiting for them to leave, and I'm going to accept the other team as soon as they are here. Judges, the team's not here, so I'm trying to get a hold of them. Okay.
Okay, good luck.
So judges, the team is not here yet. We are trying to get a hold of them. Meanwhile, we're going to wait because the other team is in 20 minutes. So if you want to take a bathroom break or get some water, feel free.
Okay, thank you.
Hi, Maria Jimena. Are you from Crimson Wolves?
Yes, I am. Sorry for the delay. We were having struggles which link was our
That's okay.
session.
So we will begin, but your time will be deducted, okay?
Okay.
So we will share the screen with your video first. Ale,
Sure.
are you doing it? Okay.
Let me know if it works with the sound.
Cancer, being a leading cause of death around
It's
the world,
fine.
continues to be a worldwide problem. The complexity of this disease lies in the different types of cancer there are. Crimson Wolves wants to take a step forward in cancer treatment by approaching colorectal cancer, or CRC, in Mexico.
According to the Instituto Mexicano del Seguro Social, colorectal cancer is the second deadliest cancer in Mexico, having an annual incidence of 15,000 cases and 7,000 deaths.
The reason why CRC is a latent problem is due to the available diagnostic technologies. As a developing country, Mexico is still working towards the implementation of better technologies around the diagnosis of cancer.
The present diagnostic techniques revolving around CRC are either non-specific or invasive, making it difficult to have an early diagnosis. Another issue arises with the symptomatology CRC has, because it is difficult to differentiate from other gastrointestinal diseases.
Our team, with the aid of synthetic biology, proposes a solution for a better diagnostic of CRC. The idea involves engineering of a probiotic that, in the presence of CRC biomarkers, will generate a measurable response.
The mechanism will consist in the binding of periplasmic proteins to the specific biomarkers present in the gut lumen. Once the proteins are bound to their antigen, a kill switch inside the probiotic will be activated and thus causing the probiotic lysis.
Once the probiotic is lysed, the liberation of pigments will occur. The fecal samples will need to be further analyzed in a laboratory for a better assessment of the diagnosis.
Our diagnostic technique will allow the patient to avoid a late diagnosis of CRC.
This could be the next step in CRC diagnostic technologies, not only in Mexico, but for the world as a whole.
Thank you. Judges, if you have questions, please raise your hands. Yes, Pablo.
Can go if you want. My question is, what will be the use of your proposal? Do you plan to take a pill with the modified polarities as a diagnose? And would it involve the killer switch in the same Gs, or is it a separate component?
Yeah, hi. Thank you for your question. So our proposal main function would be a diagnosis in a non-invasive way. And because of this, we use fluorescent pigments. And the whole mechanism would be inside of the gist because it would be a modified gist. So the kill switch would be in that mechanism as a whole.
So would it be in the same polarity, in the same Gs? It will be both the killer switch and also the diagnose part. Is that correct?
Yes.
Okay.
The gist would be the one with the pigments as well.
Okay, thanks. I don't have any follow-up questions in this case.
Thank you. Margareta.
So thank you, team, for exploring an important issue in diagnostics and in medicine. Have you considered how sensitive your test would be? Could you do calculations or modeling on how much of the probiotic you could use, for example, to detect biomarkers at an early stage of the disease?
Yes, thank you so much for your question. For the modeling part, of course, if we did an in silico, we could see the calculations for the specific antigen that we are using. For now, we only have this theoretical part, being these, how much specificity these, the antigens that we chose have per se, and these are around or above 60%, so they are really useful. They vary for each antigen that we chose. The one that has highest specificity, I think it was around 90%. So it varies.
Could I do a follow-up?
Yes.
I was curious about the probiotics. How much is this organism used today as a probiotics to prevent disease?
Can you hear me?
It's very low.
Okay. And nowadays, this probiotic is used to treat different diseases like diarrhea in infants, and previous tests with hematological patients. There were no cases with sepsis or negative effects.
Thank you.
Thank you. Alejandro, please.
Yes, thank you. Can you help me understand a little bit more? How did the modeling that you did help to improve and to change, if necessary, the design that you made?
Hello, can you restructure the question, please?
Yes. So you have designed the genetic circuit, and my question is, how did if you did the modeling of the circuit, how did this model and the simulations that you can get from there help you to improve the design or modify the design if necessary?
Sadly, our team members that were in the part in the modification and in the part of the team that were modifying the system could not be here present, but it was a process since seeing the parts that sometimes those parts were not beneficial for health, and so then we need to change those for something that were beneficial for our project, and those things took a lot of time.
If I may add, our modeling process basically was with the benchling and the simulation of the circuit of Tinkercell.
Okay, thank you.
Do you have a follow-up question? Okay, Nico, please.
Hey, yeah, my question is, so you mentioned that you plan to use fluorescent pigments, right? I'm curious if other diagnostic people. So, like there's been some recent papers that have, there's companies and lots of people trying to develop diagnostics for other things, not necessarily colorectal cancer. But I'm curious if anyone else has used fluorescent pigments and, what the challenges with that might be. So, if these pigments traffic through the GI tract, and then you have to detect them at the end, what equipment might you need? Are there other options for detection that you think would be better? How did you arrive at these pigments specifically?
Yeah, actually, it was a big of a challenge, this part of the project, because we needed to be able to measure the pigments afterwards. And basically, we are going to do this with spectrophotometry techniques. And because these pigments show high spectrophotometry specificity, and we arrived to this conclusion in a very bumpy ride because our initial idea was very different. We didn't use, or we didn't thought of pigments until later on, because we were thinking just to use binding of molecules per se, but it was easier for the methodology to have pigments involved. Also, one of the challenges would be the gut permeability. And so we chose our pigments with low permeability so that it won't go through the gut lumen.
Do you have a follow-up question, Nico?
No, I think that makes sense.
Okay, thank you. Andres? You're on mute.
Sorry. Why did you choose work with GIS, and why with Saccharomyces or not instead or Pichia, for example? As you know, maybe the cell wall of GIS is a difficult structure. So in this case, I choose GIS instead of.
I can answer this question. I think the question is, why did we use this chassis and not a more common chassis, like, I don't know, like E. coli? Maybe because the microenvironment of the tumor, it's anaerobic, and also the pH decreases per se, like from a 6.4 to a 5.5 pH. And this yeast strain is anaerobic, non-pathogenic, and this probiotic agent works well in low pH.
Thank you.
Thank you.
Do you have a follow-up question, Andres?
No, thank you.
Okay, thank you. Do any other judges have any questions?
We have a question on the chat, so if you want to answer that, go on.
Can you read it, please, Sally?
Yes. That's long. Let me open the chat. Is there a probability of affecting the balance of the intestinal microbiome, especially with I don't know how to read that, as an antifungal chitinin? And how do you ensure the degradation of the antifungal chitinase? And that word, sorry.
We have already solved this concern with a kill switch, and there is a minimal probability of having sorry, I forgot the word. There's something called fungemia, but the studies have that have been made indicate that despite the colonization of many of these organisms, there were no problem with onco patients. So there should not be any problem.
Thank you. Any other questions, judges? Yes, Pablo.
Yes. Do you know the regulation in Mexico about these kind of like samples? What would you expect if eventually this could happen in real life? What would be like the challenge that you might face?
In order to have this project to come to life, we would have to do several experiments, not only with modeling and in silico, but also we need to use in vitro techniques. We need to see the microbiology part because we are working with biological system. We would need several steps in order to be approved at the end.
I think I did the question round then. I was more expecting if you can use samples coming from humans or if you have received something about that.
Oh, like having a cancer cell culture and doing the project there. Yeah, of course, we could do that, and it would be one of the parts of the project. But I think there also comes some problems with using cell culture lines, and that would be also need to be approached.
Okay, thank you. Well, we have 1 minute left, so I'm going to stop you right here. Thank you very much for your participation here, both the team and the judges. So team, I will ask you to please leave the room and follow us on YouTube to see the other teams. And we will wait for the other team to appear. Thank you.
Thank you very much, and thank you for your questions.
Bye, thank you.
Once again, we are waiting for the team. So again, if you want to take a bathroom break or get some water, that's okay. Team is joining now. Good morning, UNMSM. So team UNMSM is here, right?
Yes, my friends are going to enter in a few moments.
Okay. Very well, the team is here. So we are going to play your promotional video first, and then we are going to go to the questions of the judges.
Pasco es la capital minera del Perú. En el 2018 se notificó que el 58% del total de casos por exposición a metales pesados correspondían a este departamento. Aquí existe un elevado riesgo de exposición e intoxicación crónica a metales como plomo, arsénico, mercurio y cadmio. Se ha identificado que los niños entre 0 y 11 años son los más afectados. Además, muchas personas fallecen intoxicadas. Es por ello que hemos diseñado una terapia empleando herramientas moleculares y biología sintética, llamada Capsulec. Una cápsula viajará hasta el estómago para liberar a Capsulec, el cual es una mini cápsula que lleva proteínas para liberarlas en el duodeno y de esta manera atrapar el plomo para posteriormente eliminarlo. Capsulec atrapará el plomo antes de que sea absorbido y enviado a la sangre. Es por ello que nuestra plataforma terapéutica promete ser novedosa, eficaz y de fácil acceso para todas las personas intoxicadas.
Thank you, Vale. Judges, do you have questions? Please raise your hands. Yes, Nico.
Hi there. Yeah, great job picking a project that is very interesting. And, you know, I think it's great to think about synthetic biology applications to address lead poisoning. I'm curious where your designed organisms, these things that capture lead, where they would function in the body, as opposed to where most lead that is ingested ends up in the body, right? So like, does lead enter the bloodstream? Does lead, you know, it seems like most lead would maybe not be in the GI tract, like some of it would be absorbed throughout the body. So I'm just curious how these capsules, you know, where they would function, and if you could comment on that.
Okay, hi, I'm going to be the traductor in this case, so I'm going to tell my friends the question in Spanish, they will answer, and then I will translate it, okay?
Are you a student or a main advisor?
Oh, I'm a member of the team.
Okay, go ahead.
Ok, entonces está preguntando cómo es que nuestra cápsula va a funcionar en el cuerpo, si es que podríamos comentar más sobre el proceso.
Ya, para explicar cómo la cápsula funciona, es importante primero explicar cómo entra el plomo, ¿no? Tal como mencionó el juez, el plomo primero ingresa por la vía gastrointestinal al ser consumido o en forma de polvo al ser absorbido por las mucosas, ¿no? Traduce esa parte.
Okay, so to answer your question, we will first need to address how lead enters to the body. And the way lead enters to a body is via oral. So people in Pasco eat food that is poisoned with lead.
Una vez el plomo entra al cuerpo, es absorbido por vía justamente en la sección del duodeno, la primera sección del intestino. Y es ahí donde nuestra proteína funciona, ya que la proteína tiene la capacidad de capturar naturalmente metales pesados.
Okay, so once lead is ingested, it will go to a section in the intestine called the duoden, and our protein has an effect on this part of the body. So our protein will act in this space, in the duoden.
Ya muchos estudios han hablado sobre la captura que tiene la metalatoinina 1A sobre el plomo. Entonces, lo que nosotros nos hemos centrado es en hacer que la proteína MT1A tenga la capacidad de producirse en grandes cantidades para bajar su coste y ubicarla justamente en la sección donde tendría mayor capacidad de atrapar el plomo y por tanto tendría mayor efectividad.
Okay, so in this case, the protein mesallothionein has an effect on lead. And this is already been discovered and everything. So in this case, what we are doing in the project is making this mesallothionein 1A to be produced in big quantities in order to lower the cost and also to make it work in this particular section, the duoden, so that it has more effectivity.
El mecanismo por el cual atrapa es debido a que tiene una alta afinidad por un gran conjunto de iones metálicos, en particular los que tienen iones con carga 2+ y 3+.
Okay, so the way this mesallothionein acts is that it has a great affinity for metallic ions, particularly the ones with 2+ and 3+.
Thank you, team. Nico, do you have a follow-up question?
No, sounds great.
Thank you. Margareta, please.
Thank you, team. I have to say I was very happy with your presentation and the project presentation. I thought it had good structure, lots of good information, and I appreciated that it was all in English. So it made me understand the project really well. And I was also impressed by the work that you have done in human practices, but also in the science part. So I have several questions, but maybe I could start asking about the metallothionein protein that you would like to express in the duodenum. Do you think there might be any problems expressing the metallothionein in large amounts in the duodenum in human beings? Do you foresee any health issues that could be connected to this overexpression?
Ok, está preguntando sobre la metallothioneína, si es que nosotros pensamos que puede existir algún problema si es que esa proteína se expresa en grandes cantidades en el duodeno.
Can you please turn on your camera, team, just so we can see you? Thank you.
Sí, yo voy a responder esta pregunta. Nosotros también nos hicimos la misma pregunta en un inicio del proyecto y hemos tomado varios factores en cuenta en relación a la cantidad de metallothioneina que va a haber en el intestino, ¿no? Uno de estos es que no tenga ningún tipo de interacción con otras proteínas, ¿no? Por eso es que realizamos como un escaneo de las posibles proteínas con las que podría interactuar en esta porción.
Okay, so we had this same question. We were also wondering maybe that expressing too much metallothionein could be harmful for the body. But in order to address this, we took into account a lot of factors regarding how much metallothionein the body can take. And so we make our research in order we made our research to know if there is any other protein that can interact with the methylthionine in the duodenum, and we verified that our protein, methylthionine 1A, doesn't interact with any of them.
Prosigue. Dentro de todo este scan que se realizó para poder ver si es que interaccionaba o no, encontramos solamente una proteína que ya estaba reportada experimentalmente, que era la megalina, que tenía una región específica de interacción con nuestra proteína. Por lo que procedimos a realizar una modificación en esta región de la proteína. Por suerte, la región en la que interaccionaba con la megalina era una región linker que no que no estaba relacionada en las zonas de interacción con los metales pesados. Entonces, la modificación no iba a tener ningún efecto en contra de la unión a metales.
Okay, so during our research, we did found one protein that interacted with methylthionine, and this one is called megalin. But the thing is that the section of this protein megalin that interacted with our methylthionine was a linker. So it wasn't, let's say, so it didn't have any effect on in regards of heavy metals. So the modifications we did on our methylthionine didn't have any effect on this.
Finalmente, para verificar que de verdad esta modificación, digamos, que tuviera una función y que en realidad no interaccione con la megalina, realizamos un docking proteína-proteína previo y posterior a la interacción para evaluar cómo es que esta modificación iba a afectar, si de verdad funcionaban. Encontramos al final que posterior a la modificación, la probabilidad de interacción con la megalina en los mejores modelos del docking era demasiado baja, y en los últimos modelos del docking era nula, ¿no? Finalmente vimos las zonas en las que el docking los modelos del docking nos daban, ¿no? Y evaluamos que no era nada similar a lo que se había detallado experimentalmente, por lo que al final concluimos que nuestra modificación fue exitosa y no iba a tener ninguna interacción con la megalina, ¿no? Por lo que al final nuestra proteína en el intestino no iba a interaccionar con ninguna proteína, y al mismo hecho de que vaya al intestino iba a terminar, digamos, pasando por todo el tracto digestivo sin interaccionar y siendo prácticamente inocuo, ¿no?
Okay, so after that, we tried to assess that our modification in the methylthionine didn't have any interaction with megalin. In order to do that, we realized molecular docking, and we found out that before and after the interaction, before and after the docking, there were no interaction with megalin. So in this case, we managed to succeed in making the methylthionine not to interact with any other proteins, even with megalin.
Can I do a short follow-up?
Yes.
Yes, I'm curious also if you did some modeling on the stability of the modified metallothionein that you had designed.
Sí, realizamos el proceso de la parte de modelamiento estructural. Fue el modelamiento, realizar la minimización de energía, ¿no? Hacer una dinámica estructural para observar cómo es que la proteína se comportaba, digamos, en el entorno, ¿no? Cómo es que iba a tener ese comportamiento y cuál iba a ser la mínima energía necesaria. Y finalmente realizamos el docking con la proteína ya, digamos que con la proteína post dinámica molecular, ¿no?
Yes, in this case, we did the structural modeling. So what we did there, we tried to minimize the energy and to see how this modified metallothionein would interact in the environment. And for that, as I said, we searched for the minimum energy, and the docking was done after the protein being minimized regarding the energy.
So you envision the protein will be stable when expressed in the human?
Pregunta si es que entonces si estar seguro, bueno, si es que estamos seguros de que la proteína va a ser estable en el cuerpo.
Ya, para responder esto, se hizo el modelado de la sección del duodeno en el tiempo, ¿no? Se hizo el modelado de, primero, cómo la mucosa va a cambiar en el tiempo, segundo, cómo la concentración de iones de plomo va a cambiar en el tiempo, y tercero, cómo la concentración de nuestra metallothioneina va a cambiar en el tiempo.
Yes, in order to do this, we did a modeling in the duoden regarding the time. So we wanted to see how the lead concentrations will change in time in the duoden. And we proved that yes, this modified metallothionein is stable in the human body.
Ya, espera. Entonces, luego de hacer este modelado, lo que encontramos fue que, pese a haber un flujo intestinal constante, un flujo de mucosa constante, se llegaba a un equilibrio en los niveles de metallothioneina más aproximadamente a las 3 horas.
And in this work that we were doing, we found that the metallothionein equilibrium is reached at 3 hours.
Y es por esto que las cápsulas tendrían que tomarse más o menos cada 4, 6 u 8 horas. Sobre todo, hay una ventana de tiempo importante que es durante el consumo de los alimentos, donde se produce la mayor cantidad de entrada de plomo al cuerpo.
And this is how we discovered that in order for our capsule to work, this will need to be taken every 4, 6 to 8 hours.
Great. Thank you very much for answering that.
Thank you. Alejandro, please.
Yes, thank you, guys. So I have a lot of questions, but we are short on time, so I will select the ones that I think are more interesting. The first one is a follow-up to this. How you will be sure how are you going to be sure that the metallothionein will be present, and is connected to Nico's question at the beginning, that will be in the place in the duoden to where the highest concentration of lead is happening? How will you keep the protein there if there is a flow of, like you said before, if there is a flow in the gastrointestinal tract, how you keep the protein there to be working as you expect?
Ya, esa pregunta es muy interesante porque al momento de modelar, claramente, la proteína no se puede mantener fija en un lugar. Así que simplemente modelamos cuánta cantidad de proteína iba a estar en ese punto específico y cuánto plomo iba a haber en ese punto específico.
Okay, but
primero hay que esperar que traduzca Jorge, y voy a seguir respondiendo.
Okay.
Ya, un momento, para volver a repetir. Lo que hicimos fue ver cuánta proteína iba a estar en el duodeno y cuánto plomo iba a estar en el duodeno, ya que hay un flujo constante de recambio ahí.
Okay, so first we wanted to address how much protein will be in the duoden, because as you said, there is a constant flow in the gastrointestinal tract.
Ya, bueno. Entonces, una vez pudimos modelar cuánto plomo iba a estar y cuánta metallothioneina iba a estar, hubo un punto particular con el cual nos aseguramos de que al menos la metallothioneina se libere en ese punto específico. Y para liberarla ahí, usamos un proyecto anterior del iGEM que tenía una membrana, una cápsula con una membrana capaz de anclarse al duodeno.
Okay, so once we discovered this, that there is a gastrointestinal flow, we found that there is a previous work in iGEM in which the group used a membrane that was able to attach in the duoden. So we based on this in order to make our mesallothionein to be kept in the duoden.
El flujo que arrastra las proteínas también arrastra el plomo. Y dado que el proceso de interacción de metallothioneínas y plomo es prácticamente instantáneo, en picosegundos, más o menos 10 a la menos 9 segundos, 10 a la menos 12 segundos, asumimos que era un proceso instantáneo, por lo cual, mientras mantengamos concentraciones superiores de proteínas a las de plomo en esa sección, estaríamos asegurando que no va a haber plomo libre, o al menos en muy bajas cantidades.
Okay. So can I have a small follow-up?
Sí, por favor.
Yes.
So it's regarding the modeling. It's nice that you use the toggle switch, which is the fundamental circuit in Synbio, like from 2000. Can you explain very briefly how you use the modeling to improve the toggle switch? And more importantly, why did you select the toggle switch to drive the production of the metallothionein?
Claro, primero con el por qué. El motivo por el cual se usó el toggle switch es porque nos permitía tener la capacidad de encender y apagar el circuito. ¿Por qué es importante? Porque la metallothioneina, al acumularse en la célula, es tóxica, por lo que si se hubiera encendido, mataría a toda la población celular.
I'll ask them to translate the answer, please, so the other judges can understand.
Okay, so as for the first question, why we use a toggle switch, is because the toggle switch allows us to keep the complex in on state and off state. And we did this because the metallothionein 1A is toxic if it is kept in the body. So having this ability to be able to switch it off, it's really important for us.
Ya, y ahora, el cómo se mejoró esto es debido a un estudio que medía cuán estable es el toggle switch bajo diferentes condiciones. Y estas condiciones eran la potencia del promotor y la capacidad transcripcional.
Okay, and as for how, it's because we review a study in which it was measured how the toggle switch acted under the, let's say, the power of the promoter and also the transcriptional capacity of this.
Entonces, teniendo estas variables que iban a afectar los estados finales, los estados estacionarios, los state states, fue que nosotros pudimos modificarlos poco a poco con tal de tener esa capacidad de que el circuito sea estable en estado prendido y en estado apagado, sin afectar la producción de proteína.
Okay, so taking these variables into account that might affect the steady state, on and off state, we were able to modify them in order to have the capacity of it being stable in both on and off states without affecting the capacity of producing the protein.
Adicionalmente, el toggle switch nos permitió aislar el proceso de activación y desactivación, haciendo que sea completamente ajeno a la producción de la proteína, por lo que este sería un proceso apartado y por tanto podríamos modificar toda la producción de la proteína y lo que lleva sin afectar ya la estabilidad del toggle switch.
And once we did this, it was very important because it became, the production of the protein became kind of an isolated step in which we were able to modify it without affecting the production capacity.
Great, thank you.
Thank you. Margareta, did you give up?
A short
for one quick question.
Well, maybe Pablo should go then. I already asked a question.
It was a really short question, and it's just out of curiosity. What does your mini robot or the satellite from your presentation video means? Does it have any special meaning?
Estoy preguntando sobre el robot androide que hemos usado en la presentación, si es que tiene algún significado o cómo, bueno, cómo es que se nos ocurrió.
Bueno, el cohete espacial fue idea del grupo de multimedia y la idea que tenían era que hacer más sencilla la explicación, ¿no? De cómo iba a ser llevada la cápsula, debido a que la cápsula iba a tener las proteínas, entonces la idea era graficar la cápsula y cómo iba a llevar hacia un lugar desconocido estas proteínas. Pero sería más interesante que el grupo de multimedia pueda explicar esa pregunta, si hay tiempo, ¿no?
Yo le podría responder. La idea principal, como menciona Diego, es hacerlo más interactivo. Y en este caso, si ven en nuestros pósters, el astronauta sería la cápsula que ingerimos. Eh, en teoría, este astronauta llevaría dentro de sí las proteínas y las minicápsulas, ¿no? Entonces, por eso también en el video se ve cómo lo va llevando. Es una representación más simple.
Please translate.
Okay, so the main idea of this, let's say robot, first is to make it more interactive, and because the astronaut is kind of the capsule, and we see how this astronaut carries all the proteins, and you can see it on the video, how the astronaut carries the proteins to unknown places, in this case will be the duoden.
Thank you very much. Well, thank you, team. Thank you, judges. Your time is up. So I'm going to ask you to please leave the room so we can have the other team.
Thank you.
The other team's already appearing, but if you want to grab some water, that's okay. We were going to wait just a couple of minutes. Now we have Ollin Synbio. Ollin, is everyone from your team here or will someone else arrive? Hi, Ollin team. Is everyone here now? We have a lot of students today.
Yes, we're a lot of people.
That's great. Okay, so I'm going to ask you to, if you have not renamed yourself yet, please do so. And also keep your cameras on when answering the judges. We are going to display your presentation video, your trailer video, and then we are going to go to the questions. Thank you, Ollin. Judges, if you have any questions, please raise your hands. Yes, Pablo.
Well, first, congratulations. Actually, I like the way you handled your project. It was like consistent and simple. So that's really nice to see. And it was well documented, so it was easy to go through your page. My question is related in how would it be, what would it be your advantage against other methods that are already in the market and also other similar methods with this kind of proof that are in, in academy?
I can answer that. Well, first of all, thank you for all your kind words. And I think the advantage that Diana has against other, it's first, the application of these aptamers gives us a large road to go ahead in the terms of giving it a lot more of possible molecules to detect. As the system functions with the principle of an aptamer in there, not a specific aptamer in there, we can just add more aptamers that that have affinities to different molecules, and thus give it a lot more use for our system. Other things I can say is that as we treat it as a local problem, various of these devices are not, we are not able to purchase them in Mexico due to, for example, regulations of importation of those materials. But these aptamers lack a really advanced regulation here in Mexico. That is something maybe after my HP area can, our HP area can talk you about. But yeah, we managed to make a really simple design that can be improved a lot more with a little bit more work, and that is usable here in our vicinity.
Can I do a follow-up?
Yes, you may.
Okay, awesome, great. And now that the competition is over, I would like to know if you think that you made the right decision picking Aptamers, and if you have more time and if you have the resources, would you change something? Would you do something different?
I can answer that just so you follow. Yeah, I think we made the right decisions with Aptamers. It's a really basic and easy to work system. I think the things we could improve a lot more are those more aptamers that we can obtain from practical from laboratory tests, because as we are in Design League, is really the in silico processes of aptamer obtention are not really mature yet, so we couldn't really rely on that, and it's really just something that gets done in the laboratory. We can also make a lot more tests to be even more sure that our device works in the conditions that we are aiming to use it. Like, for example, DNase test to see how much DNase are there in commonly consumed beverages, to ensure our system will stay consistent in those conditions, and a lot more tests that can be done just to ensure that our project can stay strong in that environmental conditions.
Thank you.
Thank you. Margareta, please.
Thank you, team, for the project description and your ideas for solving this problem. Maybe you can explain a little bit more about the available kits on the market. Are those based on aptamers? And if I may, aptamers, I think, is probably a good choice. I would envision that they're quite cheap, but the fluorophores might not be. So have you done any economic evaluation of your product? Thank you.
I would like to answer this question. Of course we did. First, we did a market analysis, and we found 3 possible competitors. One of them is actually not available in the market. We don't know why it was retired, but we found other 2. It's Centus, if I remember correctly, and Nosum. And the biggest problem we found is that they are very expensive because they are now in research stage, and they are not currently available. Besides, they don't detect multiple drugs as Diana does. And we make a big table with all the costs Diana will take. For example, the place where we are going to produce them, materials we need, and of course the most complicated part was the aptamer synthesis. But as we plan our business model, we hope return of investment in 3 years or something like it. And that's why that's how we done all the research and investigation behind all Diana implementation to the market.
Thank you. A quick follow-up. Did you do any prototyping of your proposed product? Is it a simple straw, or do you envision some other parts of your product, Diana?
team?
Yeah, I can answer a bit more of that. Yeah, we envision it as a simple straw just to maintain the simplicity and the easiness of use and transportation of the device. But we would like to add more methods of signal interpretation on this device to ensure that every person can get a clear signal of this system and can make good use of it. For example, the signal that we have in the current model is only visual, but there was some work done during the year to see if it was feasible to add other kinds of signals, like pairing it with a Bluetooth transmitter to give a vibration into, for example, a bracelet that the person has the change in the system.
A quick follow-up, it's qualitative answer, not quantitative, right?
Yes, that's right.
Thank you.
Thank you. Alejandro, please.
Thank you guys for the project. It's really interesting, the approach you took. I would like to know a bit more how did you use the information that you got from the modeling to improve your design, and specifically I saw you did the statistical mechanic work, which is really interesting. Because FRET is a very difficult technique. I don't know if you have tried in the lab to make it work, but it's really dependent on the distance between the two fluorophores and the environmental conditions, pH, and everything that will be around in the glass. So how can you assess if the system will work or not with your modeling? Thank you.
Yes, the mathematical modeling itself does not really take into account the system. It was more for us a method to see which of the characteristics of the system were the more important ones. We arrived to the conclusion that the distance between the initial distance between these two fluorophores was the main thing we should take in mind while designing and while refining this system. As you said, I read quite a bit about FRET and how difficult it is to work to get the right distance, to not get initially a false positive for say, but to also ensure that they are sufficient, they are close enough to once the signal you're trying to get, the change you're trying to get is correctly presented in the FRET system. So yeah, it was basically that we wanted to make sure which of these characteristics was the one we should take more into account, and it was basically the distance between our two fluoroproteins.
Okay, thanks.
Thank you. Any other? Yes, Margareta, please.
I'm just curious whether there is any data on how the assay will work in liquids containing alcohol and quite high alcohol levels, I would presume. Do you know?
Yes, we actually take that into account to do the molecular docking. We try to mirror the conditions that the system will be used on. So we we searched for commonly consumed beverages, the more commonly consumed the most commonly consumed beverages in our country, to see first the alcohol concentrations and also the salt concentrations of these, because these two parameters can affect a lot the how the DNA behaves. So the point at which the DNA is starting to be heavily affected by alcohol, it's around 70% concentration, but no commonly consumed beverage reaches those high levels. So we are not really worried about the effect of that on our system.
Thank you. Judges, do you have any other questions? Yes, Pablo.
Yes, my question is more related to the scaling up of these. I'm not familiar how aptamers are currently being produced for the market right now. So did you take a look in these, or how would you plan to produce like these aptamers in big scale?
I can answer this question. Actually, yes, we are looking to make it more scalable. So because we are looking that the device is accessible for everyone. But yes, we are still looking for how we can do this implementation into of synthesis. Well, all the creation of the device, because it will go into an industrial level. So then we can go delivery to all around here in Mexico City, and then all around the country.
I don't have any.
Thank you. Do you have? Oh, okay. Alejandro, please. No? Okay.
Yeah, no, I was talking with me. Thank you. Now changing gears to the human practice part. I like your different approaches that you had to try to solve the local problem. Can you tell a little bit more on how the interactions that you had with your stakeholders made the project to be like it is now?
Yes, I can answer that question. First, we have an exhaustive investigation for the problematic here in Mexico City. So we have the age for the stakeholders. Then we found that local, we have the necessity to give them information about this problem. So we made a guide for centros nocturnos. So in this guide we did investigations about how to know these that are with problems your beverages. So and then this guy bueno, then about después de la guía nosotros hicimos algunas pláticas en escuelas cercanas a nuestra ciudad. En este caso hicimos una actividad de concientización en una escuela secundaria. Hicimos esto porque
Magali, si quieres, puedo traducir.
Ah, okay.
I can traduce the answer.
Yes,
okay.
Please
translate.
Yeah,
well, I'm gonna translate. I'm sorry. I'm gonna take on where you left off, and then we can follow up. Okay. So we actually noticed there was a huge necessity for information, not just like the device, but we needed the teenagers, the people that were going to be one of our primary stakeholders needed information. They wanted to know more about, well, they wanted to live the experience, but they also needed information. So
I'm sorry, you're an instructor, you cannot translate. But if you want
I'm sorry,
to ask,
I chose the wrong,
okay.
background.
Go ahead,
I'm not an instructor. I just chose, I was informed later, but I'm really sorry. I'm part of the team as well. But yeah, sorry. So yeah, we developed the nightclub guide, and we wanted to reach our stakeholders in a friendly manner. And we also developed some activities in high schools that were one of the ages we detected that they wanted to live these experiences, but they had zero information about it, or they were one of the targets for this problem. Magali, ahora sí, si quieres continuar y sigue traduciendo.
Bueno, como ya habíamos comentado, hicimos estos acercamientos porque supimos que el consumo de bebidas empezaba desde antes de los 18 años en localmente. Por ello hicimos estas pláticas desde jóvenes, que es como tercer año de secundaria, y así le empezamos a hablar a los stakeholders.
Yeah, with all of the research we performed, we noticed that teenagers in Mexico start drinking before the age of 18 years old. So that was our target audience. No sé si quieres agregar algo más, Magali, de la parte de riesgos. Okay.
I'd like to complement what Magali and Mariana just said. Apart from what they just said of the approachment they did with schools and the stakeholders, we also made an approachment directly with nightclubs in the environment the device would be used on, mainly to do this proof of concept and to get this feedback in order to also better the design and do better iterations of the product in the future. So with this, with this information, we came up with this, we got this information for the better, for a better design for the device. And we also noticed that there was certain groups that were vulnerated, and that we also had the opportunity to generate resources in order to cover those the needs they had in these environments. So we did this approachment with nightclubs, and we got a lot of information and feedback from them in order to improve on our device and to generate other resources and attack other vulnerable groups.
Just a quick follow-up, can I, Christian? So can you specifically tell me what you did? What did they say that made you change something, and what was that?
Okay, yes. With this approachment, mainly with the nightclubs, we got a lot of feedback, but mainly they talk about the methods and the ways you could identify an adulterated beverage. They normally, and the people that are in charge of these nightclubs, they normally have como la maña, ¿cómo se dice maña? The wit to identify, and they have their methods to identify adulterated beverages. But when we presented this proposal of another method to identify these adulterated beverages, they were very interested in the functioning. They were very interested in using and proving, not proving, and in using and, ¿cómo se dice? Probar, using and testing the device once it was finished. So we got that information from them, and that also led us to an opportunity to later go and test our device with the environment, the main environment of our problematic.
Thank you.
Okay, thank you.
Guys, our time is up. Thank you very much for attending. I will ask the team to please leave. I'm going to again say thank you to all the judges. Your work has been amazing this morning or afternoon, depending where you are. If you could stay in the room just a little bit, I would like to talk to you. And team, if you could please leave, that would be great. Thank you very much.
Thank you. Have a nice day.